Okay, just a moment. Okay. Mm. Okay. Good morning, everyone. Every dignitaries in the panel, and to my dear batchmates and juniors. So, and good afternoon from Philippines. So, thank you everyone for your presence. So, this is about my dissertation or master's research presentation, and the
Akash,
topic goes. Yes.
just a minute. Would you share your video?
Okay. Sorry. Uh, is it needed? Okay.
Yes, yes, we did.
Okay, sir. Just a moment. I turned on the camera. I don't know. Is it taking time? Is it okay now?
Can't see your face. There is no video of you.
Okay, sir. Just a moment. Is it showing now?
Yeah, yeah, we can see you.
Okay, sir. Uh, okay, so if everyone is ready, then we are good to go now.
Yes.
Yes, carry on.
What about your screen?
Share your
Oh
screen.
no. Sorry. And when we talk about Asia, we of course talk about rice. So China and India being the major producer and as well as the countries who produces which consumes most amount of rice, and with respect to Philippines, they are also not left behind. They are also ahead of like major developing countries like Japan and Brazil in terms of consumption and production. And not only the production, like if you have nutrients, like on the basis of which nutrient you need and in which crop, you will introduce that nutrient into that particular crop. And this can be achieved through agronomic practices, conventional breeding, and yes, the advanced biotechnological tools such as genetic engineering and genome editing. So yeah, this question may come your mind like why we need biofortification and more micronutrients. And yeah, like you all know, basically undoubtedly very much essential for our body and for the growth of the children and other developmental stages. Iron is a very important micronutrient because it is needed for the physical and cognitive development. And for women, basically for women during their pregnancy, like there are a lot of issues with iron, so they need iron very much. And in rice, as you can see, whenever you'll go from rough rice to brown rice, then brown rice to polished rice, the content of iron decreases. Yeah, just like 8.8 ppm to like 4.1 because of the removal of the aileron layer. Okay, so now we are talking about the genome editing over conventional approaches. Why I will choose genome editing and not the conventional approaches. So to defend this, I will say that it requires less time, and we don't have to apply large amount of chemicals or like other fertilizers maybe, and stable development of improved lines. If we can introduce the gene and make it as a homozygous line, then it can result in proper establishment of the gene. So here CRISPR comes in the picture because this is one of the most revolutionary discoveries in the field of genetic engineering, and like it has totally changed the game of genetic engineering. And why someone will prefer CRISPR? Because it is more efficient than zinc finger nuclease, which was earlier used for like minute genome editing. And with CRISPR, you can also develop non-GMO products, like it will be called as cisgenomes. And yes, the main thing is specific mutagenesis, which can be performed by CRISPR, which can't be performed earlier with the normal transgenic methods. And it is also very much helpful in gene insertions. Now I'll just clear you my objectives. So basically, my work has been divided into two parts. You can see the first part is the evolutionary study of the NAS gene families, and I will go for several studies. There you can see. I'll come up in the presentation later on. And the second part is the screening for the OSNAS2 gene, which has been inserted in the UTR region, UTR of OSNAS3. And that will be done by various three methods. So just to clarify in the beginning, I'll say the first part of my work is bioinformatics work, which requires softwares and soft skills in a good laptop or in a good computer. That's what you need. And the second part, you need a lab to perform the tasks. So without any further ado, I'll go straight forward to my work. And yeah, these are the review of literature, which actually help. I'm not going to dictate them one by one. These are actually the literature, which are the major sources or inspiration of the work. And some of from my lab also, like this Jack Mito et al., they worked on this OSNAS genes. This scientist is from my lab only, and they started the work 2016 actually. And these are also the papers. So why NAS genes? Like you can ask me, like why I have targeted NAS genes? Because NAS genes produces this compound, the S-adenosyl L-methionine, and this compound is somehow used in a bioavailability of iron. S-adenosyl L-methionine is not only used in iron, but it is used for many things. Yeah, but bioavailability of iron is a major thing for the S-adenosyl L-methionine, which is also called as SAM. And there are mainly three genes, OSNAS genes, nicotinamide synthase genes in the oryza sativa, and these are OSNAS 1, 2, and 3. And as you can see, that actually gramineous family plants and non-gramineous plants, they both have different metabolic pathways to produce the S-adenosyl methionine and finally producing in mugenic acid, which is actually considered as the precursor of the iron bioavailability. Yeah, so why we need evolutionary study? In the previous thing, I told you why NAS genes, now why evolutionary study? Evolutionary study are important just to know the fundamentals of evolutionary forces, how worked on them, like how evolution changed the genes of same family or genes of other families, like how evolution worked on them. We can see the pattern and we can get some insights of them. So for the evolutionary study, we have to do multiple sequence alignment, which is aligning the sequence as much as sequence you want and see the comparisons within the sequence. Like this is an example of how multiple sequence alignment basically looks like. And a phylogenetic tree. So a phylogenetic tree is basically gives you a relationship within the genes. Like you have many genes and it will give the relationship, like which gene is related to which gene. And the main thing, the conserved domain analysis. If you have many protein sequences, obviously the genes ultimately transcribe and then translate into proteins. So in the protein sequences, you need to know which are the conserved areas. Why conserved areas? Because conserved areas were not changed from the very beginning when the evolutionary forces started working, or like changed very little. Okay, so as I was told to briefly discuss about CRISPR-Cas9 system, so I am going to do that. I'm not going to go on so deep, but it will be really precise and all the informations are here. So if the easy guide to CRISPR-Cas9 will be just two components. You divide it in two components. One is the guide RNA and another is the Cas9 protein. So the Cas9 protein is basically an endonuclease. It will cut your DNA, and the guide RNA is a 17 to 24 nucleotide sized synthesized RNA, which will be designed by you. Means you will control the design. You will design it, and then it will bind to your target sequence in the genome of any organism, and then the Cas9 protein, it will go there, make a scaffold, a ribonucleoprotein complex, then it will cut it. You can see here, like if this is your guide RNA, this is your guide RNA, and this is your genome which is attached here, and then like the Cas9 protein will come and it will just cut in this sequence and make a double stranded break. This image has not been downloaded. It is been created by me, and I have also kept it in my thesis. So basically, this is prepared by me using the Adobe Illustrator software. So what happens when the DNA has been like cut, a double stranded break? So basically, the DNA repair pathway opens, and there are two major things: homology, directed repair, and non-homologous end joining. So as you can see, the TSB has made, and then the cell can go either choose for non-homologous end joining or homology directed recombination. But the actual thing is, cell never chooses homology directed recombination most of the time. They mainly goes for the non-homologous end joining. And in my work, we are also gone for non-homologous end joining, which means the DNA is cut, the cell will repair itself. Means something will be added in this place. Something. You don't know what is that something, but something will be added, or something may get deleted also. You also don't know that. And homology directed recombination, this is a user directed thing. You can control in which place you want to cut, like knock in the sequence or knock out the sequence. Okay, just to simplify it, like there is the cut and you want to add a specific sequence like ATG you want to add. You can add ATG with the use of homology arms. But in case of non-homologous end joining, you can't add ATG here because you don't know what is going to be added by the cell there. Okay, so basically with CRISPR, you can target your sequence, you can knock it down, and sometimes you can use it for insertion, which is interesting in my work. And okay, I'll like to tell about IR64 rice variety also, but yeah, as you people will be knowing much more than me, but still IR64 has been developed by IRI. And the interesting thing about this is like IR64 inherits the same HD1 allele, and which was ultimately derived from DG Wugen, and it also has the loss of function of early heading date and the heading date 1 gene. And few things about IR64 are like in Bhutan it was released at FKR42, and Cambodia, China as NIP 11, and Indonesia, Vietnam, and rest of the African countries, Gambia and Mozambique as OM29. And this was my targeted rice, and this is about the CRISPR vector which has been transferred. So basically, for making a CRISPR reaction, you need to prepare a vector. A vector is actually a modified plasmid circular DNA, which will be transformed by agrobacterium, and then it will go and cut inside your cells. So the transform CRISPR vector was created by combining 3-module vectors, which were ordered from the Whitehouse lab in Minnesota. Okay, these are not necessary, to be honest. But the main things are single guide RNAs. Yes, basically everyone uses one single guide RNA, but we are using 2 single guide RNAs, and it has Cas9 coding gene, HPH gene, which is also called as HPT, but formally in document it is told that we should call it as HPH. And the donor OS-NAS2 gene, because we are going to donate OS-NAS2 gene inside the UTR region of OS-NAS3. So there will be expression of 2 genes, which will help in increasing the iron content in the IR64 rice, and the promoters for these genes, and nucleoplasmin NLS, which is needed for activating the nuclear localization signal. This is the vector actually, and you can see this is the Cas9 gene, and this is the donor OS2, and yeah, these are all the others. I hope like you to read a vector map, you need to start from a place and then you can go in one particular order. And these are the restriction enzyme sites which are used to create the destination vector. Basically, Golden Gate assembly method was used, and yeah, basically that's not the main part of my work, so I'll not discuss it here now. But I need to clarify the principle to you. So the principle is, as I told you, 2 sgRNAs. Like this is one sgRNA going to bind here. This is another sgRNA which is going to bind here. This is 1 and 3, not 1 and 2, because these were our candidate sgRNAs. There were 5 sgRNAs and only 1 and 3 worked. So that's why 1 and 3. And this is the UTR of OS-NAS3 gene. So it will cut here, expected. Okay, so it will cut here, and this OS-NAS2 gene will be fitting in this place. So the main thing is, here is one promoter, here is the OS-NAS3 gene, sorry for the bad drawing, and then here is this gene if it is successfully expressed. So there will be expression of OS-NAS3 here and then OS-NAS2 here. So this is our expected thing. The OS-NAS2 gene will be incorporated in this particular area. Why we are targeting UTR? Because we know that there is no such functions of UTR till now. Till now it is not discovered. So we can knock out this portion. It will not harm the plant or harm the function of the OS-NAS3 gene, and we can introduce another gene in that place. Okay, so basically the principle is done. Now we are going to like go straight forward to the actual work. Okay, so for getting the NAS genes, I used the multiple sequence alignment. And for getting the multiple sequence alignment, I need to collect the sequence. So there are OS-NAS1, 2, and 3 in rice, but there are other NAS genes in other monocot plants too, like wheat, barley. Then we also searched in other Oryza land races like Rufipogon, Brachyantha, Longistaminata. So we searched in the database called Grameen Ensemble Plants and maize GDB, which is the official database for maize. We selected the sequence, we took the only best identical percentage results, then we gave them a proper name, and then we again blasted them and collected the sequence. And these are mainly the sequence initials. So OS-NAS means it's derived from Oryza sativa, Rufi means Rufipogon, Bart means Barthi, and so on, like for Sordby, for sorghum. Okay. Then after the multiple sequence alignment, I did the phylogenetic analysis of the NAS genes, like all the collected NAS genes. And this is the software interface. The software which was used is called as Mega. And this Mega software is useful for doing this kind of molecular evolutionary studies. So basically, the phylogenetic tree was performed by neighbor joining algorithm and with 1,000 replications with bootstrap analysis. This is basically a method which is like, it's easy to perform. Then I did the concept domain analysis. So first the multiple sequence alignment, then the relationship by using phylogenetic tree. Then I will do the conserved domain analysis. So here is a catch here. So basically, I used another database which is called as motifsearch.genomenet, which is used to search any conserved domain for proteins, and the domains were and the like the protein sequences were represented as these blocks. Suppose I have one protein sequence. The protein sequence of OSNAS1 is like, just say it's 300 amino acid. So to represent 300 amino acids, it will be too long, right? It will go like this, and it will look bad in the presentation or in the thesis, or mainly in the documentation. So basically what we do, we used one block, this one block as 60 amino acid. So this one block will denote as the length of 60 amino acid. And this one block will also include the domain size and also the subdomains. This will represent the particular things. So now we are done with the material methods for the bioinformatics part. Now we'll come to my lab work part. So as the OSNAS2 has already been transferred in the OSNAS3, so we need to design primers to detect the OSNAS2. And to detect it, I designed the primers based on the amplicon size, because the thing is, as you know, the gene was inserted in a blunt cut. The gene was inserted as a blunt cut. So if this is the gene and this cut is blunter, this is supposed to insert here. So the gene can rotate 180 degree and can join in that place, and also can flip and join. So if you design the primers on the basis of the OSNAS2 sequence, you are not sure that in which orientation it has been inserted, and you are not also sure about your results. You need to develop many primers, so that will maybe use a lot of resources and money. So that's why we have designed the primers on the basis of amplicon. Means on the basis of the size, we can at least tell, okay, our gene of interest is there. So the OSNAS2 gene size is 2424 base pair long. So if we get a band of 2.4 kb, we are sure that there has been something inserted at least. So the primers were created on the basis of the flanking regions, like this area and this area. So the one primer will start from here, and the another primer will start from here. And basically they will amplify something if there is something. And if there is not something, obviously it will not amplify. So we can say that, yeah, there is nothing there. So I'll not dictate and like what I did exactly. So basically I used Primer 3 Plus software, and I checked if there is any here, which is very necessary. Also the GC percentage and repetitive sequences are not there. And I used OligoCalc to check the herp information. And also they were blasted to see like if there is another matching thing there within the same chromosome or not, so that it will bind there also and it will amplify it also. Okay. And this is the genomic DNA isolation. So I started with isolating the genome edited IR64 rice. So I extracted the DNA, and the thing is, we just used a modified method. Everything is same, a bit of modifications, not so much. Something in portions, and like nothing to tell so much about it. Yeah. So basically, I started first screening the Cas9 and HPH for the amplification. So these are my marker genes. So if these genes are present, I am sure that the OSNAS2 also may be present. If they are not present, then I am sure that the OSNAS2 is also not present. And I had like 600 samples in the beginning. Actually, I had 1,500 samples, but it was reduced to 600 samples for the pandemic. So, 600 samples or like these are too much. So we need to discard them first. So for discarding, for primary selection, we did the Cas9 and HPH selection. So these were my protocols. So yeah, that would dictate so much about it. And kappa polymerase was used. I'm dictating it because it is a very good DNA polymerase enzyme, my PCR never failed. Like every reaction was successful. So after screening for the Cas9 and HPH, I did the optimization for the primers which I have designed. So basically, I designed 3 forward primers and 3 reverse primers, and I had to make combinations. So 3 into 3 are 9. So basically, you can see that there are 9 combinations here. Forward primer 1, reverse primer 1, forward primer 1, reverse primer 2, so on it will go. And for that, I performed a gradient PCR because I don't know the primers are new and I don't know the annealing temperature of them. I need to select the annealing temperature. So I set the temperature from 55 degrees to 65 degrees actually. And this was done like automatically by the PCR machine when you perform a gradient PCR. So there are 12 wells, and each 12 wells in the PCR has different temperature. And my, all the primer combinations are done differently. So I prepared 9 different PCR cocktails and I performed them in this gradient PCR. So basically, my aim is to select which temperature works well for my designed primers. It can work well in 55, it can also work well in 64. I don't know. Let's see. And these are mainly the protocols which I followed. This is the how I prepared the master mix. This is the reactions cycle. And this is the final work, which is the detection of the OSNAS2, like whether the OSNAS2 has been detected or not. So for that, I all used the PCR and the design primers, and the primer which will be performing better in optimization like this of this combination, sorry, of this combinations, I will select them. Okay, so now I'll go to results and discussion. So the first thing, as you know, was the bioinformatics part, and which was finding the NAS genes. So I got 31 NAS genes candidates. They are ortholog sequences of OSNAS 1, 2, 3. So these are my main sequence of study. I blasted them in other databases and I got 6 genes like the NAS genes of GMAs, like maize family, of barley, of Luistaminata, Brachyantha, Barthi, Glaberrima, Rufipogon, sorghum, and wheat. So I got 31 genes. And then I did the multi-sequence alignment using Mega. Then I did the phylogenetic tree. So you can see this is how a phylogenetic tree looks like. So what is interesting about this is the OSNAS 1 and OSNAS 2 are similar. Okay, first I need to tell you how to a phylogenetic tree. So basically, this is the root of the phylogenetic tree, and these are the branch points. So the branch points basically diverges between 2 different types of origin. So the organisms from this branch point and the sequences from this branch points or organisms, whatever, like mainly the sequences here, are different, are actually different, and also different from this. So whenever this will be divided, it will create that they are not similar, they are different. So you can see that the KN FG006 gene is the most diverse gene, which is actually a oryza longistaminata NAS gene. So this NAS gene is actually responsible for iron, like production of the S-adenosyl methionine and somehow making more iron in the oryza longistaminata. And the gene is really, really long and very diverse from all the genes. And OSNAS2 and OSNAS1 are similar as they are in same category, same clade. I'll come to clade later. And the OSNAS 3 is a bit different from OSNAS 2. You can see it is different. And basically, the clades are like when a branch point is divided into 2 other sub branches, you can see that this branch point is divided. So this is one clade. And if by analyzing this, I got 6 clades 1, 2, 3, 4, 5, 6. And we can like divide the genes in these clades. You can ask what is the necessity. The necessity is basically if you want to do any experiment for let's say in maize, or let's say in Hodium vulgare, or in wheat. So we know to target for the iron, we know how which genes to be targeted, and even we need any landrace gene, we know that which gene to target. So yeah, this is the relationship between the genes I got, and this is the conserved domain analysis. So I got around 148 domains and 21 types of domains. So domain is basically has a different functions. So these are actually regions of protein which has a specific functions. So these are the protein sequences. Okay, just to clarify in between, all the sequences are actually protein sequences, amino acid sequences. These sequences you see, they are all protein sequences I collected, not the DNA sequences. I collected the DNA sequences, nucleus sequences, and that gave me the same tree. There are no different results. So I prefer studying proteins because with the protein sequence, you get the conserved domain because, yeah, ultimately you need the protein sequence. So it's better to study the protein sequences. So this is the example how I got the results. So I just showed you all the pictures are in my thesis. So as you can see, the OS-NAS gene has the NAS domain, and then it has this methyl transferase 25 domain, it has PAS8 domain, transferase 31 domain, and DUF domain, which is domain of unknown function. So all the 31 sequences has different domains. But what our aim is to get the common sequence, the common domain. And surprisingly, I found the common domain, actually not surprisingly, it is NAS. Yeah, every gene has the NAS domain. And surprising thing is every NAS domain had this methyl 25, methyl transferase 25 domain. So for functioning of the protein, the NAS domain is needed, and also the methyl 25 transferase, like methyl transferase 25 domain is needed. But in two cases, I didn't get the methyl transferase 25 domains. But in rest of the 29 sequences, I got all. So this is how I represented. So it is easy to read. You can see this is the amino acid sequence, this color. So this is the whole sequence length. This is the NAS domain. I only selected NAS domain because I have to study the conserved domain, right? So that's why I have selected the NAS domain. You can see this much long is the NAS domain. And this is the methyl transferase 25. So this is here. So this is basically a graphical representation of what result I found. You see the NAS domain is this much long, and within the NAS domain, the methyl transferase 25 domain lies. So this is for OS-NAS1. Similar thing is done here. This is the NAS domain, and within this, the methyl transferase 25 domain lies. But let me clear you, it is not necessary that in all the proteins, the NAS domain will be in same region. You can see in this sequence, here is the NAS domain, there is another NAS domain here. Here is no methyl transferase 25, but there is methyl transferase 25 in this domain. So these are the subdomains inside the domain, but the main domain is NAS domain. Okay. So these are the studies for the all 31 sequences you can see. And this is the most diverse sequence, the longest amino acid sequence I told you. It's longest. It is very long. It was like 1098 amino acid long. Okay. And there are also domains of unknown functions present, but yeah, that are not necessary to include because the functions are still unknown. So the bioinformatics part is done, and now we are off for the results for my lab work. So first was the screening of presence of Cas9, and you can see a clear gel image of PCR. The Cas9 gene is 2.1 kb, and you can see the bands are between 3000 base pair and 2000 base pair. So basically, many plants had this Cas9. You can see, and the blank is like here, so means it is not contaminated. And the aim suggests the DNA marker, the DNA ladder. This is the screening for HPH or HPT. You can see the HPH gene is 554 base pair long. It lies within 750 base pair and 500 base pair. So this is also what can I say, successful PCR. So you can see that I got many plants positive for Cas9 and HPH. So you can also think, okay, if the plants have Cas9 and HPH, both, if the plant has, just see, like you can see plant number 340 has this Cas9 and also has this HPH. So it means if it has both, it means the whole tDNA of the vector has been inserted. And if the tDNA is there, it means the OSNAS2 is there, because the Cas9 and HPT are the border genes of the OSNAS2. It means we can expect that in these kind of samples, we should see at least any insertion of OSNAS2. Yeah, then the optimization of my PCR. So basically, these are the primers which I designed: AK45, AK46, AK47. So basically, these are the forward and the reverse primer, and then I made the combinations of forward primer 1, 1, 1, 1, 2, 1, 3, 2, 1, 2, 2, 2, 3, like this. And this is my expected band size if there is no gene insertion. And this is my expected band size if there is a gene insertion. So the result of my gradient PCR, you can see. So all the primers work, but you need to see here clearly that F1R1 didn't work well, because in 55.1 or this temperature, there is no result. There is also no result here. There is also no result here. There is also no result here, because temperature, the primers did not anneal. But in this temperature, the primers annealed actually. So these are the results of all the combinations. You can see 1, 2, 3, 4, 5, 6, 7, 8. Oh, sorry, 9. So I selected this one because you can see all the bands are clear. All the bands are present in all the temperature, and the bands are so distinct, so visible. So you can expect that these primers are working well. Yes, and the band size is between 500 to 750. This is because you can see I selected F1R3, and the F1R3 band size without an insertion will be 521. So yes, it is 20 between 750 and 500. Okay, so I selected the primer pair F1R3, and the temperature I said is 56-degree, because in 56-degree I got a very distinct band, very distinct band. Okay, so this is a supplementary data. This was before COVID. This was actually a part of my work, but due to COVID, like area is so strict. They locked down everything. I was not allowed to go in the transgenic greenhouse, nowhere. So this is before COVID. The data I got. This is called a supplementary data. I also included it in my thesis as appendix. So this is just to let you know the phenotypic or the agronomic traits. So basically, I managed to study height, and surprisingly, I studied fluctuations in the plant height. The tallest plant was 115 centimeter, and the shortest plant was 77 centimeter. But the IR64 normal value is 110 centimeter, basically, or sometimes 105. So there is fluctuations, and we think that this is because of the genome editing. There may be off-target editings, means there may be other genes which has been cut down instead of the UTR of OSNAS3. So you can see these are the controls, and they are similar. Yeah, there are no such fluctuations, but see the genome edited plants. Oh my God, so fluctuations. Yes. This is for the plant height, and this is for number of tillers. For number of tillers, the fluctuation is even more. There was a plant which had only 2 tillers, and there was another plant which has 18 tillers. The normal tiller number is, as you can see, is the control is like 14 and 16. Like we can say the freeze average. So you can see the fluctuation, right? 2, 3, 4, 5, and in some plants there is like 10. And what I discussed with my supervisor is like, yeah, that may be off-target editing, because one of my friend, he got a plant which started getting ons. That variety never had ons, but after genome editing, it got ons. Okay, so this is a bit of data. This is not exactly the part of my thesis, but yeah, that is what I studied and I showed you. And the next slide. Yeah, this is transformation. Just to let you know that I also did the transformation myself, but I did in Siherang rice in the beginning. Basically, transformation is the making the genome editing work, the first stage of genome editing work. I prepared the genome editing solution with, like, it's not genome editing solution, just to make you feel understand. I'm telling it as such way. So basically, I prepared the agrobacterium solution. Then I transferred it into the immature embryos of Siherang rice, and then I kept it for co-cultivation. So then the genes will be edited. So I didn't include in my thesis because the plants were only in the chamber. I couldn't go and see the Siharang plants, and I couldn't get the data because of lockdown. But yeah, just to let you know, I also did the transformation with proper efficiency, and it went good. Okay, now the final results, the main result for the OSNAS2 screening. So yeah, fortunately or unfortunately, I actually didn't get till now an OSNAS proper OSNAS too. But what I am calling fortunately is because the gene got edited. Why? Because if you get bands between 750 and 500, that means there is no OSNAS2 genes. There is nothing inserted. But surprisingly, in some samples, I got a band. You can see here a band between 300 to 100 base pairs. So like we thought, like, what is that band? Is it a contamination or is it something else? But there it is not a contamination, because you can see the negative control, there is nothing, and in the blank it is nothing. If it was a contamination, it was present in all the samples, at least it would have been present in blank. So what is this? And surprisingly, I got many of this kind of bands which are between 300 and 100 base pair. So basically, what is that? I will conclude it in my conclusion. So I am done with my results, and now this is for summary and conclusion. Just to briefly tell you the summary, just to tell you in short, the summary is, it is not necessary that if a gene are from the same family or same species, they will be clustered within the same clade. It can come in other clades, means in other, it may be diverged. Like you can see they are same family, Osnas 1, 2, and 3, but they are different. 1 and 2 are similar, but 3 was different. And yeah, methyl transferase 25 is present in all the genes except in these two genes. And NAS domains are present in all the NAS genes. So it is clear that for the function of NAS gene, you need to have the NAS domain and also the methyl transferase 25 domain or the subdomain, and which makes it crucial for the function of the NAS genes. And the DOF domains, yeah, they're also important because they're unknown functions. Maybe they have some functions, and if you study, maybe we can get to know something from that. Now about the evaluation, yeah, many samples were Cas9 and HPH positive. You saw in the PCR result, right? Many plants had Cas9 and HPH. But yeah, why did I didn't get any OSNAS2? Like, because maybe the thing may be the band is so small, there may be only one insertion, and the primers are not able to amplify that, and it's not detected. Basically, it may be an heterozygous T0 plant, like T0 plant, maybe. And yes, as I told you, I used NHAG method, not homology directed recombination. So NHAG means I will cut the gene and it will get inserted of its own, but the cell will repair itself. So the frequency is basically low. It is proven in several articles and journals that if we go for NHAG method, the gene is not inserted properly, but still we tried. That's why it's called an experiment, right? So it may be possible that in that place there might be insertion of something, maybe some part of the gene, or there may be some insertion deletions also. There may be insertion of a larger fragment, then it got deleted. So there is still something fishy or something unclear, but it is clear that we need to screen more and more plants, because the more plants we grow, The more plants we transform, there are chances of getting more, like at least one plant we can get if we maximize the screening. So what I will suggest, or we will suggest, for the future work, for the bioinformatics work, what we should do next is the homology modeling of the proteins, which is basically comparing the structural differences, structural homologies between the protein. And also we need to get to know the three-dimensional structure of the domain, like how they will exist, what is their, like we need to know how they will exist. And it is necessary to know the domain functions so that we can actually get to know the function and we can get the gene, we can select the gene we should target to biofortify any crop which is like which for which we need more iron. And for the lab work, what we will suggest, yeah, just I told that large number of sample screening, and yes, modification in the screening, and the main thing is sequencing. Yeah, so we need to send some samples, like the sample which I got, the small bands which is between 300 and 100 base pairs, to get to know exactly what happened. And it is also possible that in the gel it is not showing, or in the PCR it is not happening somehow. But if we sequence it, like the whole genome sequencing, if you do that, then we get to know that there might be the possibility of the gene which has been inserted. So sequencing is the only final way which will give us the results. But yeah, it was lockdown, you can't do it. And yes, but we will do it, the work will progress in such way. So yeah, so this is like what I did, and this is what happened. And this is the time for acknowledgement, and it is really necessary, because first I'd like to obviously thank from the bottom of my heart to Dean Sir, Professor Thomas Das Gupta Sir, for giving me the opportunity to go to IRI and conduct this, because it was needed. And to all the Maharajas, like I respect them from the deepest core of my heart, and my institution, Ramakrishna Mission, yes, I will be thankful to them. And Dr. Inez, my co-supervisor, who is the head of this genome editing or the transgenic or the molecular biology department. She is a true leader, and she's so humble. She cared for me. She supervised me in a very good way. And this research work would not be possible without Ma'am Yvonne's work. So Ma'am Yvonne is actually postdoc of my supervisor. She was always there for anything. Ma'am, this happened. Ma'am, what to do? She was always there. And a big thanks to my GTL family, like Genomic Transformation Lab family. So these are my lab mates, Dr. Ruby, Kuya, Ate. Kuya means big brother, Ate means sister in Filipino. And yes, I will be always thankful to Dr. Ajay sir, who is now the Directorate of Research of IRI. And I am very thankful and respectful to Ashutosh sir, Rambabu sir, Kishore sir, Gautam sir, Masud sir, Mukesh sir, and to my IIT Guwahati team, all my teachers. And yes, a big thanks to my friends for believing me, supporting me. And the main thing, I am nothing without my parents, my sister and her husband. Their love and belief makes me stronger. Thank you all for bearing with me through this presentation. Thank you.
Thank you, Akash. It's a good and nice presentation.
Thank you.
Slide was beautiful, neat and clear and understandable. I think you had done a lot of work during this short period of time. I
Yes,
open this
only
session.
2.5
Mhm. Is it Mukesh sir or not? They have joined lately. Mukesh sir and Rahman sir. If they are, I would like to welcome them and open the session for the comment, suggestion, question. Starting from our respected Dr. Subbu sir. Sir, any comment? Subbu sir, hello.
Yes. Shubro, start with the Shubro.
Yes, sir.
Yeah, Professor Dr. So, I think for your guidance, Mr. Akash Doctor. Was able to do the nice work, and probably I am not the fittest persons to evaluate to his thesis, because it is a very high level work. It's a very it's a very good, not only good but a high level work. And I just want to know one simple question, but I couldn't understand, and to only enrich myself, that Akash, how you can prove that OS NAS 1 and OS NAS 2 is similar, but OS NAS 3 is more diverse? This is my one question. And
Yes, sir.
this is one question. And my second question is, in this way, how much iron percentage can be enriched in the right state?
Okay, sir. Thank you for the questions. So I just like to confirm the question. Your first question was like OSNAS 1 and 2 genes are similar, but OSNAS.
How you can ascertain?
How we can ascertain? Yes. So I'll again go to the slide, or yeah, it's like explain. And we compare two sequences, to protein sequences are long. Sequence of like at least protein sequence will be let's say 100 amino acid long. So there are 100 amino acids like this. So we can compare it manually, but it is never manually done, right, sir? Because we have many sequences. Why we will manually? So we use soft. So this software help us in acknowledging differences. They have own programs. So you will put the sequences in the software, and then they will give you the differences. And but manually, you just need to do a multiple sequence alignment. You have to click on the consensus sequence site or conserved sequence site. From that, you can know. Like in OSNAS 1 and 2, there are some things are present in both, sir. Like some region of the, like some region which are present in, but that same region which was supposed to be present in OSNAS 3 is not present. And instead of that thing, there is something else. So that's why they are different. Sequences are not same. If the amino acid sequences are not the same, actual protein will not be same. They will be producing a different protein, and different protein means a different of secondary structure, in the tertiary structure, then the quaternary structure, and ultimately the protein will look different and the function will be different.
Okay. Okay. Just see the protein analysis. Okay. Okay.
Yes, sir. And about your second question, sir, I remember, sir, last time when I presented, you told me it will be better if I do the biochemical work. But you can see I didn't mention any biochemical data because, sir, only reason I can tell you it is obvious that it is COVID-19, sir. And what we are going to do next is we're going to collect the seeds. We already collected the seeds. I manually collected myself. And we are going to do the mass spectroscopy, or we are going to do ICP. And using this ICP method, we are going to estimate the iron content. And we are also going to estimate the cadmium content, sir, because it is also seen sometimes while editing a genome, the plants get modified or mutated, and it also absorbs the heavy metals from the plant. So it means, sir, if you are developing a plant with more iron, that doesn't mean that you are doing a good work. It can also result in increasing arsenic or cadmium in that plant. We need to like calculate the concentration of iron also, as well as cadmium, then arsenic, lead, other heavy metals too, just to be sure that the thing is going good. We may get good iron, but then if we get more cadmium, then it is also not acceptable. So
Well,
basically, the data will be collected later, sir.
thank you, sir. Uh, maybe Dean sir can ask after this one. Hello, sir.
Yes.
Yes.
Uh,
Ashutosh, asking
yes, sir.
something?
Yeah.
You first
Can you
tell Mukesh to interact, then I will interact.
I didn't see sir here. Mukesh
Huh?
sir.
Mukesh, sir.
Mukesh sir has left.
Okay. So I was just searching some seminar talk. I just heard from there is a scientist called Roviraj Banakar from Avestajan Limited Company. So who worked with the original theme of CRISPR-Cas9 scientist who developed this idea in Spain. So he worked there for several years. Now, then he was at USA, then joined in a multinational company. He was giving one seminar, a webinar seminar at BAU, that means Bihar Agriculture University Sabur. So they invited me to join there. So I was hearing same thing, genome editing thing. And one thing he told about that perhaps higher gene is involved with the low cadmium uptake. Akash told about the cadmium. So his conclusion was also on that. He did a lot of work for his whole life of 25, 30 years. He worked on genome editing and learned that CRISPR-Cas9 technology from the original founder. So that is one thing. So we have to be sure regarding cadmium and zinc, iron relationship. And secondly is that you worked on T0 plan, no? T0 plan. Yeah. So have you heard any impact after that T1, T2, T3, maybe some influences or some deteriorations or some advancement in T1 or T2? Any idea?
No, sir, because I studied the T0 plants from the beginning. I studied when they were in pre-leaf stage inside Yoshida solution. I transferred them with my hands in the pots. Then I grown them, and they were grown in as they matured in March or April, sir. Basically, in last week of March to April. Then the lockdown happens, sir. And what we are going to do, sir? Actually, we are going to study the T1. The next thing we are going to again plant them. Transgenic greenhouse, and we are going to
Sometimes there is deterioration or improvement also T1. So it is not that it is a stable one. So far my experience goes. And secondly is that the variability in the plant height, tiller number, etc. you have shown because of the effect of the insertion, isn't it? Is it true? Yeah.
yes, sir. Yes, sir. Yes, sir. That is what we are considering because we think that there might be off-target editing, sir. Means it can cut down from another gene, maybe where it got the similar sequence and it got attached and it cut that place down.
So from the plant breeder's point of view, that means your genome editing is a precision-based work. That means you want to insert the specific part in specific genome, etc., and see that. But still there is variation, isn't it? Still variation, it may be good, it may be bad,
Yes, sir.
as you have
Yes, sir. But
shown. In your work you have shown, maybe some of them may be good for the variation point of view, but it is only for the variation, may not be good for the other agronomic evaluation. But still you have done a good work. My suggestion is that you have to know the basic techniques of the creation of the, from the agrobacterium insertion, etc. That means creation of the T0 plant. That is very important. Needs a lot of efficiency in
so
doing
that's
the
what
work.
I'm doing.
That is very important.
Okay.
You know that I did some work in France in gene silencing, aquaporin gene, you know, which is important for the drought tolerance. So it needs a lot of exercise, expertise also at the basic level to learn those techniques, and then go for the other thing. And final outcome, be sure about the final outcome, because we bidders want the final outcome. It is not that you have inserted and there is variation, but we need the what is the expression at the morphological level, or actually what has happened. So for the biotechnology kind of work, or that is good, that is also important. And but we need the final outcome also, because the people will look into the, after doing, spending a lot of money as well as doing a lot of hard work, what actually happened at the final. So
that is one also.
yeah. And so you have to know the basic technique also, because I have seen in France how efficient they are in doing the transgenic plant. All the people, even the lab assistant, other people, they're very, very efficient. And also need to know the outside scientific world, what actually happened to the final outcome. Otherwise, you cannot clarify your position that what you have done. So all these things are needed. And actually, people think that it is a Shubhra kind of very sophisticated work, but if you go into the work, then you will find it just like other work they are doing.
So,
Not that something like that.
but
So,
so far as the NAS sample is concerned, sir, it is really worthwhile.
So this is important. And another important, by hearing into, because you are our student, and hearing into the, listening into the many recent conferences, etc., because of the this online. Now everything is on online, webinar or like that. So we need to, breeder need to think of the many other things, like the right from the haplotype analysis, we bidders can do, or the biotechnologist need to know what is the importance of, suppose I've heard that whenever a gene is suppose the high yield or high panicle length, etc., in rice again, but it needs a lot of time is left to actually the apply the concept, maybe 10, 20 years time. And so what my feeling is that there is a gap of the application side as well as the biotechnology work side. And also we need some bioinformatics side also. We have to learn. We have to learn. Nobody is, suppose the specialized bioinformatic people, their idea is somewhat different. And our bidder idea is to always go for the application side, what happens into the field. And biotechnology, basic fundamental biotechnology, they lie inside. So we need to learn many things. Suppose the gene concept, haplotype concept, because some of the haplotype concepts are really interesting. So,
Hello. Hello. Now it is not audible.
I think disconnected. Hello, sir.
Akash, you can hear?
Sir. Yeah, I can hear you. Then, sir, I think just
I
connect.
think sir
then,
got
then
disconnected.
this kind of hindrance
Okay.
will be broken down. Understand? Hindrance means we think that it is a kind of higher research, not that one. That can be actually broken down by the, if we start working on the, or learning on the bioinformatics side, that many data, many things are available for Akash and ED and other things people are doing. By analyzing those things, we can discover many things that which are in the applied section, and what, how we will proceed, we can learn also. So this is my feelings from the COVID-19 pandemic situation created, and many online webinar has come, and I also joined some of there to learn the things from the many stalwarts. So thank you, Akash, for giving the, actually for going into the, poking your nose into the this area. I will not say that you have learned many things. Only you have inserted your nose to learn something. So and also maybe many things are left in the. You can go for the PhD and build up some good liaison with our institute, so that always think that you belong to our institute, and how much by your learning you can contribute to the institute to educate the students or other people, and to let them know that what you have learned. And any learning is not a big thing. That means if you go, you can learn. But if you can communicate your learning, that is more benefit for the greater community. Understand, Akash?
Yes.
Yeah.
Yes, sir.
Yeah.
Thank
Yeah.
you, sir. Sir,
Thank
I have
you.
something to say, sir. So basically, you are correct, sir. For developing a transgene, minimum, sir, it was considered we need T4 generation. But what the genome editing work does is, sir, we can cut it to T2 generation. You are right, sir,
Yeah.
because I started analyzing the sample first. So basically, what I am doing in ERI, it is like I'm going like other way around. I am like showing my nose. I know because the genes have been edited and I am evaluating. So that's why, sir, I have included the bioinformatics work just to know how to get to know any candidate gene. That's why I have included this bioinformatics part. Yes, sir. With your regard and your suggestion, I have started now building the CRISPR-Cas9 constructs. Like now they are teaching me. Now I am working in the lab making the golden gate cloning and the golden gate assembly to make the transformation. How we can do. So, yeah, now I am doing that, sir, and I am learning. Let's hope, sir, someday maybe something may come out from genome editing, sir. You never know.
Thank you, sir. I would like to round up
Nice
this one.
interaction. Yeah.
Mhm.
I've learned also told many things that really actually just start to move. There is a high scope high scope in so many words and work in the in ED and all the best for your path, and you must share your work to your fellow person, fellow colleague, and student, so that you will be in right point of view.
Thank you, sir. Rambabu, sir, any suggestion?
Hello, sir. Good afternoon.
And this even in this pandemic situation. So I have done many things research work. I think I will just
Not audible.
Sorry, sir. Not,
Sir, we are not hearing you
not clearly
clearly.
audible.
sound is not coming clearly.
Rambabu is not audible.
Your
Hello, sir.
question is not clear. Listening problem.
Oh, sir, I have no question.
Oh,
Okay, sir. Now it is audible. Sir.
Audible?
yes, sir. You can speak.
Hello?
Yes.
Yes, sir. Yes.
Yes, audible.
I have no opinion. Akash, you have done very good work in very short time period, even in pandemic situation. So thank you very much, Akash. I have learned many things from your research work. Thank you very much.
Thank you, sir.
Thank you, Ramabu, sir. And I would like to ask from our teacher, sir, biotechnology. Really, they have some questions. Teacher, sir?
Yes, yes. So thank you, Akash. I think this is an excellent piece of work you have presented, and this is the kind of trend-setting work. And I would like to say our coming students, the freshers and first batch, because this is excellent training of PCR and this bioinformatics work, and we have everything. And you are also doing some of the courses like tools and technique, which are very much related to this work. So I recommend you to please nicely see the things, how it works, what are the principle of these techniques, and how can we apply in the our applied work like varietal development or purity of hybrids or something. It can be anything related to plant breeding. And Dean sir has pointed out that there are many things in plant breeding which can be done using the this molecular biotechnology or molecular breeding, we can say. So I think this is one of the very good work and nicely presented also. I have only one suggestion to Akash that while running the gel, while putting the marker, you have to put one positive samples, okay, positive control, so that we can clear. Like in case of while putting your sample, you have to put the vector so that we can get the particular bend size. Okay, so that will be nice if you could do this. And this is the one of the bridge, you can say that you are bonding, making the bridge between the breeding, plant breeding and the biotechnology. And the cutting edge science is really very interesting nowadays. So, and one thing I would like to say that you put the evolutionary insights of NAS gene, and this is very simplified mechanism you have shown that how can we utilize, how can we train the MSc student in this area. So this will give the complete and deeper understanding of the how this gene evolved from the wild species and how they are domesticated and come to the current varieties, and how the genes, sometimes genes can be lost during this domestication. So while putting this evolutionary study, we can say that how the genes are evaluated. Okay, so this is very good work. I think breeder and my colleagues should start dwelling into the evolutionary things, I think. So this is a very good example of study. Thank you, Akash, for presenting nice thing, presenting cutting edge scenario of molecular breeding. Thank you very much.
Thank you, sir. Thank you for your remarks. I'd just like to add one thing, sir. Our right, sir. We are supposed to add any positive control, but what actually happened? We didn't use the vectors. We went for the gel, and next when we get the Cas9 or HPT, we just selected the plant and then used that plant as the positive, sir. Actually, we did that. So I had one photo, sir, which had the positive. But sir, for the OSNAS2 screening, we don't have any positive, as you know, sir, because we don't know insertion, sir, because it is we are doing for the first time.
For MSc course,
Thank you, sir.
I think it's sufficient to present, although it's a nice thing. Akash, I have one confusion that from where you have cloned the NAS2 gene. What is the source of NAS2 gene?
The source is basically, if you can see, I'll show it to you.
No, which species means I want to say which.
I'm telling you, there were 3 module vectors, 3 modules. So, variety, sir. Sir, it's normal,
Yes.
sir, a normal, sir, IR72, yes, sir, but it was actually when I asked them, they're like, they got it synthesized from the company, sir, as you know, sir. They just ligated it in the vector, sir.
Okay.
All the things
Thank
you
you,
may
Akash.
not be aware.
Constructs,
I mean, all things you may not be aware in this stage because you work for a very small time, no?
yes, sir, but now the lab is open since Dean sir was saying, it would be better if the work started from the beginning. Sir, that is what is happening now. Now I am reading the constructs and doing more bioinformatics work. And yes, sir, the protein homology modeling and the 3D structure prediction, sir, that's what I am doing now. Thank you, sir.
Thank you, Akash. We are expecting your expertise after coming back to India.
Oh, I think you try to, what I told you earlier also, for your PhD degree also, you try at ED. It will be better and also communicate with us often. And maybe you can take 1 or 2 seminars here. And your guide also. That means your guide is also welcome. I expected your guide in this seminar. I thought you might be there.
Sir, actually, Madam Inez, she's already in a CGIR meeting. She asked, and I told ma'am, but ma'am, you want sick, sir. She's ill from last 2 days and she's in quarantine.
So, in any opportunity, you just invite your man to have a seminar like thing with the departments or our institute, GPB, biotechnology, etc. Students maybe he can explain in a broad way what she is doing.
Yes. Okay.
So I invite her to be in a seminar. Okay, Akash. You just tell her. Maybe sometime we can.
Sir, I can after this meeting, but it's okay. I can tell you now. The thing is, sir, you just need to send one email to my supervisor, Dr. Inez, about that my presentations that it is handed, so that they will give me the clearance for the from the ED library. So, sir, and you can invite her like sir, in that time. Let's see, sir, what her remark.
Okay.
But she is really, I think this will be possible in near future.
Yeah. I want. I do not want to lose this kind of the collaboration, and I want that this collaboration will be strengthened between ED and our IITM faculty center in future also. I told you repeatedly, whatever the way you think, you try to strengthen this relationship.
Okay. Sure, sir.
Understand, Akash?
Yes, sir.
Yeah. Thank you, Akash. Now I open the session for the student corner. Any questions from student side? No, any question? Any question from student side? Megashri? I think they have not. They have no any question. Okay, they are replying in chat also. Thank you, Akash, for the nice presentation. I hope this expertise will help us in future, our institute. Thank you, external examiner Dr. Subro sir. Join once again. You have led our candidate. Thank you, Jim sir. Valuable suggestion, remarks. And my colleague Dr. Rambabu sir and Dr. Kishore sir. Thank you, sir, for joining us here. And most of all, my dear students, thank you for joining us. I hope you could you understand something from Akash seminar. Thank you once again. Thank you.
Thank you.
Okay, sir.
Thank you, sir. Take care.